Okay, now we can start. So we have Mojgan Asadi here. So present to us the talk, the interesting talk today. Mojgan, off you go.
So thank you so much, and hey everyone. Now it's 8:06 a.m. in my time. I'm in LA. So I hope I can make an interesting talk, and feel free to reach me after actually the presentation, after the talk, so for further discussion. So one of the important questions, at least in my field, is that how artificial intelligence was beneficial for medical and biological discovery. My name is Mojgan Asadi. I'm a PhD candidate in computational biochemistry, and I'm doing dual degree. I'm doing also master in computer science in University of Southern California. My advisor is a Nobel Laureate Arieh Warshel. And so we are mainly working actually on biological system using molecular dynamic. And in some cases also, sometimes we have to answer the question using artificial intelligence. Briefly, let me just explain. When we are talking about bioinformatics, is the intersection between three different actually categories, between statistics, biology, and computer science. So it means that you should have a chemistry or biology question in order to answer question using, for example, computer. But at the same time, you need to have enough data to use a statistic in order to answer these biological questions. So bioinformatics gets many improvement and advancement for different questions, either in medicine or biological questions. For example, when we were talking about genomes, when we have a lot of sequences that we can align them, so all this data can be processed using bioinformatics and using computer knowledge. Same thing for DNA, that can be easily written down as a binary, as a 0 and 1, in order to just transform the data from a biology to a computer question. So, but today I'm going to only discuss my expertise. So we are working in our group and in my field and my experience working only with biological systems. When we are talking about biological systems, we are dealing with two main questions. What biological system we talk about? Sometimes we talk about on the right side, we talk about the biological system, a protein that is surrounded with a sphere of water. So we call it as a soluble protein. But on the left side, sometimes we talk also about the protein that they are embedded into the lipid bilayer. So that lipid bilayer can also be detergent. So these two biological systems, actually, they got so many improvement with using bioinformatics. I mean, so many questions was able to answer for using bioinformatics. And why these two types of biological questions are very important? In our body, we have many different of these proteins. So we have many different of these two types of proteins. So understanding how we can use computer science, how we can use the language we are learning in computer to addressing the problem human health and human disease have, like the disease we have, it's very important. So the project I want to talk today, I want to mainly discuss about these two types of proteins. So one of them is that when you're talking about enzyme, so I'm going to really briefly explain what enzyme does. In human body, not even human, in nature, we use enzyme in order to enhance any catalytic activity. If we have a reaction that enzyme is not involved, usually goes slower. But when we have it into the enzyme, the reaction happen into the enzyme environment, the reaction always happen way faster compared to when the enzyme is not present. So enzyme, understanding the enzyme, how the enzyme function, how the enzyme work, it's a very important question. The first project I want to discuss, that is one of my recent paper that is accepted and it's going to be released soon, is that can we use molecular dynamic and bioinformatics to address one of the most important questions, which is inhibitor design and how the inhibitor works. what inhibitor is. Inhibitor is the ligand similar to this, the blue one, that is going to bind to the enzyme and lead the enzyme to not work anymore, to prevent the function of this specific enzyme. So understanding of how this inhibitor function, like what is the catalytic activity, is so important for designing new inhibitor or new drug that can have a less side effect in human body, for example. So in my project, we work on Bruton kinase, which call it BTK, and the other one is interleukin-2 inducible T cell kinase. So this is the left side. The blue one is actually A inhibitor, and A panel is actually the superimposed of these two different kinases. One of them which is BTK, one of them is ITK. That BTK shows with the yellow and ITK shows with the light purple. So we want to see, since these type enzymes are coming from TK family, we have to understand how evolution helped, that these two enzymes, that they come from same family, still they have a bonding site. Bonding site means that where ligand or inhibitor can dock, can be different from each other. How evolution did help to make these two enzymes that they come from same family be different from one to each other. And more important question is how these differences that evolution led to can be more beneficial. So this is a very important question. So first thing we did was that we have this, by the way, this inhibitor, which is in the left side, the blue one, it's already formal FDA approved inhibitor. So it's already it's in the industry. So we just use this structure, we dock it into the enzyme, and we try to do calculation based on this complex inhibitor and its enzyme. And we can see inhibitor and enzyme here. And we can see from panel B, the differences between the residues that they are in the neighboring of this ligand. So it means that evolution will lead to changes of the residues amino acid differences to the neighboring of this inhibitor. So one of the questions we tried to answer in the beginning of our project was that, can we use, so none of this question was possible to even answer if there wasn't using bioinformatics, or for example using Python, or using only MD. Only certain questions are capable of, we are capable of answering certain questions, just because it becomes too expensive calculation. So for example, because these enzymes are coming from TK family, TK means tyrosine kinase family, we did sequence alignment. We got sequences from each one of these enzymes, and to find out evolutionary how these enzymes change over time and which residue, which amino acid they change over time. We use this task with using the tool they call it multiple sequence alignment, MSA, which is a very popular method in biology, and it helped a lot for aligning different sequences of enzymes from the same family, of the different enzymes from the same family. And that's tell us a lot that how different enzyme, for example, this enzyme, their residue and everything that changed, why it changed, and what was the driving force for that changes, and are they really, these changes are beneficial for human body? Okay. So we try to capture also for this enzyme, we have many different inhibitors that already they have FDA approved or FDA approved. So here on the left, I try to show different inhibitor, ibrutinib, ceftriaxone, zanubrutinib, acalabrutinib, and tirabrutinib. So those are different inhibitors that they are already known for tyrosine kinases for enzymes. And those are the tyrosine kinases. On the x-axis, we have BMX, TXEG, and then TEG, and different type of inhibitor that we have. And we try to show here with the pink circle how each inhibitor, it's more favorable, or has in biology, we call it IC50, means that affinity for different type of kinases. So if we understand how each inhibitor has a different affinity for different kinases, then we can understand that which exactly a structure it's more favorable for that bonding site of this enzyme. And here we try to also understand using the hydrophobicity and charge. So each amino acids that we have in bonding site of this inhibitor, they have a certain property. Either they are hydrophobic, like they are made of CH2, CH3, or they're hydrophilic. They made of like O, oxygen, nitrogen, those type of atoms that give hydrophobicity or charge to the cavity. So it's very important to understand what is really causing, what is a really important reason of this functionality of this a specific inhibitor in the bonding site. So here, this tree on the right is exactly tree of the TK family, and we try to show how BTK as a respect to their K effective was more favorable compared to ITK. And here, actually, I'm going to explain it here. So in our group, that's my advisor is very well known for specifically for many things, but for specifically for using empirical valence bond calculation. What does that mean? See, when we are trying to answer, this is always my point, when we try to answer always biological question, you cannot only, sometimes the question are too difficult, that you cannot only use bioinformatics to answer the question. We have to use also MD or even quantum mechanical type calculation to combine it with bioinformatics to answer the question, to be capable to address the question. So the question is so complicated that bioinformatics might not be good enough, because we know bioinformatics has its own disadvantage, which I will explain it later. So here we try to use also quantum mechanic type calculation. When we are dealing with enzyme, it's so important to be able to calculate the reaction that is as close as to human body. In human body, everything moves very fast. So in calculation with computer, with coding, we're using human power, right? So it's so important to be capable, to be able to design a mechanism or write down or run a calculation that is as close as possible to human body. That's why sometimes when it comes to enzyme, we use quantum mechanic type calculation, because quantum mechanic type calculation, they're fast calculation. And here, also, in order to understand the mechanism of how this inhibitor, when it comes to bonding site, how really behave, how this inhibitor really react with this neighboring environment, how it reacts that makes to this enzyme to prevent from functioning. So understanding that also needs to understand and learn quantum mechanics to every step, as when the inhibitor come to the bonding site, all the way to this enzyme can be captured with energy. What is the energy at every step that this enzyme will get inhibited? So there are several steps. So when this inhibitor docked, we have a proton transfer, we have nucleophilic attack, we have tautomerization, which I don't want to go detail because they're too much organic chemistry. But I just want to say that we need to have also knowledge of quantum mechanics, because it's so important in the level of enzyme inhibition to understand in detail, energy-wise, what is the inhibition process and catalytic mechanism. So the scheme I have on the left, it's actually for the paper that I just said it, that is coming out soon. It exactly shows panel, scheme on left, A and B, shows exactly how two different enzymes, which is very interesting, that they come from same family, but how they can behave and can energetically have different barriers. And that's why sometimes when we use certain drug, it works for one enzyme, but it doesn't really work for the other enzyme. Or when we are using inhibitor, it really inhibits one enzyme, but it doesn't really inhibit the other enzyme. That's actually the energy barrier shows, it can give us good explanation of why sometimes certain inhibitor cannot inhibit one enzyme, but can inhibit the other enzyme. And the energy plots on the right, also A and B, is exactly respect to scheme A and B. There is energy profile for every step. And all of them, they're calculated by high-level ab initio, which is exactly, and then QM calculation, which is quantum mechanic type calculation. So this one was understanding when it comes to soluble. If you remember, we said, I try to, this is for, I said beginning that we have two types of protein. So this question was addressed for that type of protein when we have sphere of the water. But let's say that if really we can do also something when we are dealing with the membrane, when we have a protein that is embedded into the lipid bilayer. Now I'm going to start I'm going to explain a new project, which was my first paper, which is, they call it, please feel free to read it, is the effect of environmental factor of catalytic activity. So when you're dealing with the membrane proteins, it becomes more challenging because always when the number of the complex increases, the number of atoms increases, always the calculation becomes more complicated. When we have a membrane involved, actually the schema the figure I try to illustrate here is the upside is the proteins in the lipid. I try to, just as a cartoon to just present it. And the bottom side is in the detergent. So when crystallization, the crystal experimentalist people, they solve the protein, they can solve it in two different, protein can be solved in two different ways and then pure it and then crystallize it. Either is a lipid bilayer or detergent. The bottom is a detergent, the top is lipid bilayer. And here in the green, we're trying to show how when the ligand moves from outside, which we call it extracellular, all the way to the last figure, which is the bonding pocket, how this conformation is going to change, and what is the role of lipid here. So what lipid can do when this substrate move from all the way from extracellular, even sometimes intracellular, depend to the system, to the bonding pocket. So we try to also using Python to capture this conformational change. So when this ligand green move from one side to the other side, we have a conformational change of our protein. So protein can have a briefly conformational change that this green, which is ligand, can bind to this bonding pocket. So we try to use renormalization method to capture this conformational change. So the conformational change that happened to the two different, as a function of substrate movement and as a function of conformation change. So we are moving from one conformation to the other conformation. So again, I will be very, feel free to read the paper and we can chat even further about this paper. Here also the other thing that currently I'm doing is that when another membrane protein, actually, when we are dealing with human bestrophin. So human bestrophin is one protein, is a membrane protein that is very important for this, for eye disease. It's also a membrane protein, but let me just tell you what's the problem here. The color, so red color is a protein, right? But the other color I show here, cyan, actually, it shows the mutations we have. And if any of these mutations happen, we will have an eye problem. So we will have a retinal disease. The number of these mutations is 250. It's very difficult to use molecular dynamic calculation to capture what is the effect of each individual mutation on human disease. So we need to use Python, we need to use coding, we need to use bioinformatics when it comes also to this type of question. You cannot go over each 250 mutation one by one to understand what is the effect of each mutation. It becomes very expensive. Expensive, I mean, it becomes very long calculation. Here, now I'm still working on it. We try to understand if we have a mutation on the neck. I'm trying to show it in the middle. Let me actually put my highlight. Yeah, so here. So this is the neck. We tried, because neck is very important, where the calcium ion bonds in this protein. We try to also find out how each mutation would participate for ion movement, because this is an ion gated channel. It means that ion move from one side to the other side. So we try to understand if we do a certain mutation take place in nature, if it's going to affect the ion movement. So this is also what we are doing. So this structure also has two different conformations, has open and closed conformation, and we try to address exactly how the ion moves. So if this is ion, how the ion move from one side to the other side when this mutation on the neck is taking place. So, of course, bioinformatics, knowing language have a very big contribution to, especially when we are comparing genetic, for genetic data, and for our calculation. And honestly, I didn't present one of my work, which I knew there's not enough time. There is another work that bioinformatics, it's so much now getting advancement, and it's actually a hot topic right now. It's the enzyme design. So enzyme design or protein design is enzyme is protein, but not all the time. But for enzyme design now, using bioinformatics gets very very popular right now to design a new enzyme. Whatever our body has a certain functionality, right? Has a certain rate for the reaction to take place. But can we even design in our body a better enzyme that can function even better? So that is a very hot topic right now. And of course, for drug discovery also, for example, the drug I was explaining, can we design a better drug that has a less side effect in human body? And those are the advantage. But disadvantage is that I think that's the biggest problem with bioinformatics. You need always a lot of data, and not always we have a lot of data. Experimentally, we need a lot of data, and that is one of the very disadvantage, that not for all the questions we have enough data. And so that's why it makes it not always we can use bioinformatics. So being said, so this is my Twitter, this is my GitHub, and this is my Google Scholar. I would be more than happy to chat if you want to work with each other. And if there is something that currently have a couple projects, if you guys want to be involved. So yeah, that's all. So I will be happy to answer any question.
Thank you, Mojgan. It was a very interesting topic, domain field for at least for me. We do have one question. I think more question is coming in. So you mentioned about the bioinformatic, the tools, right? Are there any particular Python packages that you use on this project, or is it is the tools about coding, or
yeah, it's a coding tool. So
okay.
Yeah, it's a coding tool, but of course we always have for example, a multi-sequence alignment, it has its own package, but I try to put most of them on GitHub, but mostly are just coding. But there are very interesting actually bioinformatic packages out there for biology question. But for my specific project, we always use our own codes. Yeah.
Is it would you mind, I'm just curious, is it in what language it is? Just
okay,
curious.
So no, that's a good question. So our group mainly used Fortran, but since I'm new generation, I don't know Fortran. I use Python. So
Okay.
yeah, so the language I know, and I know how to code, and even read it fast is Python. But our group and the software even we have, and sometimes we have to make implementation over that, is a Fortran. So honestly, I think if you know Python very well, working with Fortran, even let's say you don't know Fortran, it make it slower. For example, read the code, debug it, or implement it, but still you're capable of doing it if you know Python very well. So
Interesting, interesting. All right, thank you. So we have a second question here. This is a very, very common and yet important one. Are you collecting the data on your own during the research studies, or do you use like open data set?
So usually so for our field, we are usually trying to collect the data from experimentalists who worked, for example, on this, let's say, for example, this slide now that shows on this family of enzymes. And honestly, there are not so many. So always we collect them by ourselves. But sometimes when, for example, for this project, I was lucky enough that the experimentalists, they send, I'm sorry, not for this project, for Enzyme Design project, we were lucky enough that experimentalists, they send us their whole data. So yeah, sometimes we collect ourselves, sometimes no, we have to get from experimentalists, but mainly we work with experimentalist people. So we collect the data from experimentalists. Yeah.
Interesting, interesting. All right. So another question. So you mentioned eye disease mutations for degenerative eye health.
Yeah.
So this person wants to know if these studies can help determine from a single ethnic population that has traveled, if this eye degeneration can be tracked through time.
So let me make sure I understand the question.
Okay, so let me see. Let me see this. So it's about the eye disease mutations.
Yeah.
I would say okay, I think there are two questions, I would assume. So if these studies, like whether these studies can help determine from a single ethnic population. So
Okay.
it's just data sample from a single ethnic population.
Yeah. So let me just say something about bestrophin. Bestrophin is actually, so bestrophin, if you a little bit study, you will see we have human bestrophin, we have bacterial bestrophin, we have a chicken bestrophin. It's coming from different species. The question we're trying to answer, I hope I can answer the question of the whoever asked that question. But in the project, the question we're trying to answer is that we know from experimentalists we have 250 different mutations. Okay. They have a hypothesis that each mutation, where it's located, for example, if the mutation happened in the neck that I'm showing here, or look at here, or the mutation if it happens here in the aperture, they can have a higher impact or higher risk on the disease. This is not possible to capture if you don't know current. It means that if you don't know conductivity, how certain mutation can lead ion movement. If this mutation happened, really, if ion moving from one side to other side is going to change. To answer your question is that we need to know this. Understanding for all different bestrophins. So not only human bestrophin that we are working on now. So I believe I hope I'm answering your question. I believe to address your question is that we need to know each one of these mutations, how it's going to have a high impact on different also bestrophin, and all of them we have, and then get a probability of probability of saying that, oh, if the mutation happened in the aperture, this is the, for example, result that we have. So it's not easy to say if you're only discussing about human dystrophy.
Cool. So whoever wrote the question, if you need a follow-up, feel free to post it again. It's good, it's good, it's good. It's perfect. I think we have a couple more interesting questions coming up. Joy asks, so how do you manage to juggle a master in computer science and a PhD in computational biology? So she has some common background, but sorry, they both have common background, but they really need a lot of time to be master of each other.
24/7. But I was planning to finish PhD in 4 years, but I got some visa problem, like not visa, green card things, and then I have to extend it for 5 years. But I don't know. Honestly, for me, I really enjoy it, especially when you're talking about human, like something related to human health. I really enjoy it. So of course it's difficult, but I enjoy it so much. And the reason my undergrad was experimental. So I did in Columbia University, I did train to work experimental. And I said in my PhD, I want to, extend that knowledge. I want to go for computational. So I really enjoy it. And so it's, even though I work a lot, but it's not, to me, it doesn't sound like a struggling. I enjoy it so much.
It's your passion.
Yeah, absolutely. I know it sounds cliche, but you really need to have it because it's difficult. Honestly speaking, it's not easy. So many challenges. When you publish your paper, so many criticizing your result, criticizing your hypothesis, criticizing your approach. So you have to be passionate. You have to be very, very trust yourself. Yeah.
Makes sense. Okay. So you're definitely an expert to help us demystify the different concepts this attendee asked. So what is the difference between bioinformatics and bioinformatics and bioforensics?
I don't think so I can answer this question.
Oh, okay.
Yeah.
Okay, it's there's so many specific terms we sometimes did we say that wrong? It's
Exactly, no, I prefer to a little bit go research before answering this question to make sure I'm right with what I'm saying. So
okay, makes sense. Fair.
Because I don't want to say it if it's especially for bio. What was the last one? What was?
Bioforensics, forensics.
No. Yeah, please.
Okay, no problem. No worries. Okay, so do you need to be good at math to study bioinformatics?
Absolutely. Math, I think, honestly, if I one day become faculty, that's the only thing I do care. The grades for math, I think you need to be good in math. Let me just say it. I really believe if you are good in math, if you have that type of mindset, it's learning new things become easier. I really believe that. It's my personal belief. Okay, I'm not saying I'm right, but it's personal belief. I think when someone is good in math, learning bioinformatics for this person become easier because it's a statistic, it's probability. So when you are getting certain probability plot, you have to have that type of mindset to analyze it by yourself. So what is outlier? If you remove that outlier, what is going to happen? What's the significant amount of probability? What is p-value? What is n-value? So I really believe having a good math is important. And sometimes some countries we came from in high school, they teach us math in very different level. So sometimes that it makes sense that some people, they have a different level of math. But absolutely, I think math, it's very important. So that's why if you're in the early stage in your career, if you're high school, if you're undergrad, take math classes as many as you can. Trust me on that. Even when I come to PhD, they advise us that if you can't take math classes, because we don't need so many math classes, but still, if you take, they are not saying don't take it. So still they advise us take math classes.
I'm just curious, you did mention it's more statistical for math, it's more statistical related, right? It's more for statistics. Do you have any, like we call pure math, I would believe, or calculus, like solving equations, like do you have like some element on it?
Yeah, it's calculus. It's calculus that you can
Okay.
study with derivations, you can study probabilities. So it's a calculus, absolutely. So, just make yourself uncomfortable. I know sometimes math classes makes us uncomfortable. I think that is a good thing. To me, anything that makes us uncomfortable, I like it. So make you uncomfortable. So, but yeah, it's a very good direction to move.
That's a good quote. Make yourself very uncomfortable. Like
No, because
that's good.
It's true. Yeah, it's true.
That's good. That's good. That's true. That's true. Okay. We have attendee asking, can we mix synthetic enzymes to help stop mutations?
I'm sorry. Say it again.
Can we mix synthetic enzymes like to help mutations?
Absolutely, very good question. Absolutely, that is the so we are computational. We are hoping all the time when we do mutation. Let's say that we do as we use quantum, let's say, and bioinformatics and any of the methods we have, tools we have, and we understand that with certain mutation we can make this barrier. For example, I'm just giving an example, lower. It means that enzyme function faster. Okay, we are hoping we send that understanding to experimentalists to synthesize this enzyme with the mutation we are saying, that they are really make that enzyme to come to reality. Absolutely, yes. So, but always we have challenges to find experimentalist collaboration for synthesizing enzyme. But that is something that we are always dreaming and hoping to have. When we design it, they can bring it to reality. So, because we design it with computer, right? We model it with computer. But those are experimental people in the wet lab that they can bring these enzymes to become a real enzyme. And we send it hopefully, for example, for FDA proof. Yeah.
Thank you. Thank you for answering this question. Okay, we have a couple minutes left. Anyone has any more questions? Oh, one more. Okay, I was gonna answer success impression. Thank you for bringing it up. So what is your advice to young people who want to study bioinformatics, but who we don't know where to start or how to start?
So if you're undergrad, so this either your high school or undergrad, let's say. If you're high school, I really suggest you to don't narrow yourself down. Don't say, oh, I want to go only bioinformatics. It will change over time. Your interest might change over time. Even though now you're 100% sure, I'm talking about if you're high school. So even though now you're 100% sure you want to go for bioinformatics, but your interest might change. So try to learn science, especially math, physics, pure knowledge, biology, chemistry with good grades, and learn from the people who are good in your field, the people who are in university or in your school. Just try to learn. Approach PhD people at different schools. Just ask them, can I talk to you? Can I just do Zoom? I want to ask a question. Don't be shy, try to approach people, try to learn as much as you can. But if you're in college, and let's say that you come to this conclusion that you want to go bioinformatics, I really think that try to learn first. So don't jump to, oh, I want to be expert, because try to build up this career very well from base. Like, what is base? is that you know your knowledge, you have the knowledge, your knowledge is that you know one language very well, and you know the science good enough, right? You know algorithms good enough. So try to start from pure good knowledge of science, and then coding language, learn one language very well, and learn mathematics also. I really suggest you for plotting, for those type of any, I think I found a very good skill to have mathematics also. And so have few very good skill and be good at it. And then based on that, try to still, learn from people, try to see other opportunities, like other directions, not jump from one direction to other direction, but still try to be open-minded, because bioinformatics, it's a very big field, right? It's not only biology and medicine. It's not like, oh, if you know bioinformatics, you can answer all the questions. It's also good to have other knowledges, like quantum mechanics knowledge also. So don't be only expert in bioinformatics, okay? So try to be also, especially if you want to go to medicine and biology for it's for this type of question, try to also learn quantum mechanics type calculations, also empirical valence band type calculation, a little bit of molecular dynamic type calculation. It makes you a stronger candidate in bioinformatics.
That's a good advice. It's the world is really changing, and
Absolutely.
they interact into each other. Like honestly, like each field is like there's no like just think like, oh, I'm narrowing down this field. It's gonna be fine. Like it's changing a lot nowadays.
Absolutely.
All right. Thank you so much, Mojgan, for
Absolutely.
the wonderful presentation. So if you're interested in Mojgan's work, please follow her on GitHub and Google Scholar. And just a little reminder on behalf of Women Who Code. So if this is the first time, remember to join us on Slack. We are on the main social media as well, and we have some exciting upcoming events. So please register before you forget.
Thank you.
And also, please let us know. So Stephanie just sent out a link. So if you have any thoughts about on topics like anything, please let us know. Just click the link and fill out the form, and we will definitely read it and take it as a consideration for sure. All right. Thank you so much, guys.
Yeah,
thank you.
It was so nice. Yeah, thank you so much for having me.